So we are starting chapter 9, which is physical and chemical control of microbes. So we're going to be talking about how we can control the growth of these microorganisms. In other words, we want to inhibit them. We want to keep them from growing. And there are different levels of control that we're going to be talking about. So this is the all-important terms for this chapter here. So you really need to have these terms down packed, what sterilization, disinfection, decontamination, and antisepsis refer to. So these are all different levels of control. So sterilization refers to the highest level of control. In other words, this is the cleanest form. There are no microorganisms that are present on something that has been sterilized. So it says no microbes. This includes viruses. There's no viruses that are present. And the endospores are also removed as well. Okay, so we're leaving no microorganisms behind after sterilization. Now, the methods to sterilize things are not going to be suitable for living things, right? Because we're getting rid of bacteria, something that is alive. So a lot of times you cannot sterilize something that is living. It's going to end up killing the cells of the organism. So this is mainly for inanimate objects, sterilization. The next level is what we call disinfection. So when you disinfect something, the microorganisms are killed and the viruses are killed, but there's still the possibility there could be endospores left behind. So it's not as high of a level as sterilization, but it's still pretty good, right? And once again, this is usually something that you're not going to do on living tissues. So it's still going to be kind of toxic to them or harmful to living tissues. So once again, mainly for inanimate objects. Then as we step down from that, we have decontamination, or another term for that is sanitization. So we're going to remove microbes to a safe level. So that means we're leaving some behind. They're not all going to be killed. And as it says there, it does not kill viruses. So this is mainly what we talk about in the food industry, right? So when you run your dishes through the dishwasher, all right, that is sanitization. So microorganisms can still be present, but you're not eating a plate full of bacteria, right? And the food too itself, right? When you buy packaged food, it has to go through a process to reduce the level of microbes. And then we have antisepsis and degermination. So these are methods that help to reduce the pathogens that are present on living tissues. So this is how we can kind of reduce the microbial load on living tissues without harming the tissues themselves. So that's antisepsis or degermination. Degermination a lot of times refers to just kind of washing your hands with soap and the physical action of rubbing your hands together under the water with the soap, helping to remove the microorganisms. So this is the chart from your book. So it talks about sterilization, disinfection, just like we were talking about. So under the key points for sterilization, it says the term sterile should be used only in the strictest sense to refer to materials that have been subjected to the process of sterilization. So we might kind of, use it loosely in common terms, but when we're talking about it in the microbiology class, sterilization refers to that complete removal of everything. Right. And so as it says there's no such thing as being slightly sterile. All right. It's sterile. It is sterile. All right. There's nothing left behind. So once again, on inanimate objects for sterilization, it's dangerous to do that on the human body. So as it says, common usage for sterilization is mainly surgical instruments, syringes, and other things like that, right? And we'll talk about how we go about sterilizing things. Under disinfection, once again, inanimate objects used for this. Talks about using bleach solutions would be an example. of doing that are using iodine, immersing thermometers in iodine solutions between uses. Okay. And so they're giving examples of the agents that would be used for disinfection or for sterilization. And we'll talk about each of these agents as we go through this chapter. And decontamination and sanitization, right? We said important to restaurants, dairies, industries, and things like that. Okay. And there should be, I think it's kind of cut off here in this view, the antisepsis. All right, but we already mentioned that. Okay, so moving on, we've got this graphic from your book that talks about the relative resistance of different microbial types. So we have these different microorganisms from viruses through bacteria and protozoans, and it's talking about the least resistant at the bottom of our arrow to the most resistant. So we're talking about which microorganisms are resistant to different chemical control agents. So the ones that are least resistant to these agents are enveloped viruses. So enveloped viruses have a membrane around them, and it's real easy to disrupt that lipid membrane, right? Usually you use soap to get rid of oils and things on your hands. So it's so easy to, and oils are lipids, so it's really easy to disrupt lipid barriers. And then we move on to our non-enveloped viruses, to our fungi, our gram-negative bacteria, and then we get into our protozoan trophozoites, which were the active stages. A little bit more resistant are the cysts, right? They were kind of hanging out until environmental conditions get better, so they're more resistant to these microbial controls. And then we have Staphylococcus and Pseudomonas. So we're kind of, and Mycobacterium, we're giving the genera names of some microorganisms that are much more highly resistant to chemical controls. Mycobacterium has a very waxy cell wall that prevents a lot of these chemicals being able to penetrate into the cell. And Staphylococcus and Pseudomonas have a lot of different mechanisms that they use to help to kind of deactivate some of these microbial controls. And then we have at the top here the bacterial endospores as being one of the most highly resistant forms, right? We said they're just going dormant and they form this thick wall around themselves and they just hang out until conditions get better. So that's why it's kind of important that we highlight which level of control is able to remove the endospores. And remember, that's only with sterilization that you have the removal of the endospores. And then at the top here, we have the prions. We really didn't mention them. Prions are very difficult to treat. It's the Center for Disease Control that kind of advises how to treat prions. And it's usually some combination of heat and chemicals because heat alone will not kill them. So they're kind of a special group that we're not going to worry too much about. Okay. Oops. All right. So this is also a table from your book. So it's referring to these different microbial controls that we're going to be talking about in this chapter. So it refers to the physical agents in blue. So we can use heat or radiation to get rid of microorganisms. And under heat, we'll talk about dry heat and moist heat and the different methods that are used under those two categories. And you can see that dry heat provides sterilization, whereas moist heat, depending upon the method that you use, you can achieve sterilization or disinfection. Same thing with radiation. One will sterilize, the other one will disinfect. And we'll go through all of these, but this is kind of a nice way to kind of view and kind of compare the different methods. Then we'll talk about chemical agents, gases, and liquids, and the different levels that we can achieve of removing microorganisms, and then mechanical methods, which is mainly filtration, and we'll talk about filtering air or filtering liquids. Now let's also talk about a few terms before we move into these different agents. So some of these agents provide microbistatic control. Some might provide microbicidal control. So when we use the suffix of static, it refers to a steady state, kind of, right? We talk about homeostasis, right? So it's got that stay kind of ending to it. So holding steady. So it's not going to allow the microorganisms to reproduce. So if we talk about something being bacteriostatic, it's going to keep the bacteria from reproducing. So it's keeping it at its current levels, and it's not going to kind of keep on growing. Versus something that's a side is going to kill the microorganism. So if it's a bactericide, it kills the bacteria. If it's a fungicide, it kills the fungus, right? You probably hear of herbicides, right, that you might put outside to kill weeds, right? So that side refers to killing, okay? Now, when we talk about killing these microorganisms, how do we know that we've killed them? How do we know that they're dead? You can't simply look at them and say, oh yeah, it's dead. You know, it's like, it's lying down, its eyes are closed. You can't tell with a microorganism. You can't tell if it's breathing or not. So how do you know that you've been successful in killing that microorganism? So with these microorganisms, we look at whether they're capable of reproducing or not when given optimal growth conditions. All right, so if there are plenty of nutrients and it looks like it should be growing under those conditions and it's not, then we can consider it to be dead, right? Because endospores, we know, will just hang out until the conditions get good. So we want to give them optimal growth conditions, and if they're still not growing at that point, then we consider them to be dead. Okay, so loss of that reproductive capability under optimal growth conditions refers to microbial death. All right, so agents versus processes. So sometimes we'll have terms where we're talking about the agent itself, and other times we're talking about a process. So we use the word antisepsis, right, which referred to the removal of microorganisms from living tissues, and we would use an antiseptic to do that. So an antiseptic is the actual agent that is going to destroy or remove that pathogen from living tissue. All right, so it says preparing skin before surgical incisions with iodine or ordinary washing your hands with soap. All right, so the soap or the iodine would be the antiseptic. Now, asepsis refers to the keeping the area free of microorganisms. All right, so it says any practice that prevents infectious agents from gaining a foothold, that prevents infection. So A means without, so without sepsis, and sepsis refers to the growth of the microorganisms in the body and the body's response to that infection. So you might hear about somebody being septic, right, which is really dangerous. So that refers to the growth and the body's response to that infection. We'll also, when we go through these chemical agents and physical agents, we can talk about how they control the microorganisms. So on the left, you see that they can control by disrupting the cell wall, maybe disrupting the cytoplasmic membrane, or disrupting proteins synthesis, right, by targeting the ribosomes or other enzymes in metabolic pathways. Or it can prevent proteins or affect the proteins, I should say, by denaturing them. So denaturing proteins refers to the fact that proteins lose their shape. And if protein loses their shape, then they can no longer function properly. And so it shows the examples of the different agents that are on the right hand that typically work on the cell wall or denature proteins. And once again, we'll kind of revisit some of this as we talk about the various physical and chemical agents.